commencer. Bon, tout d'abord, j'aimerais, au nom du comité d'organisation de ces séminaires virtuels appelés donc AGIREmed Webinar. Donc, c'est le second de la série. Et aujourd'hui, nous avons donc l'honneur d'avoir Antonio Richard Granell du SCIC, donc que je vais présenter plus en détail tout à l'heure, qui va partager avec nous ses travaux sur la qualité et les arômes de la tomate. Donc, avant, je vais juste résumer notre action. Donc, l'idée de lancer ces webinaires pour les gens qui n'étaient pas avec nous la première fois. Donc, ces webinaires, ils ont vu le jour suite au confinement qu'on a tous connu ces derniers temps, et pour voilà, apporter quelque chose pour soutenir nos jeunes chercheurs et étudiants, et aussi pour notre communauté scientifique de partager nos résultats donc à travers notre réseau et à travers tous nos collaborateurs partout dans le monde. Alors historiquement, ce réseau et cette initiative, il est issu de partenaires qui ont avant travaillé sur un autre projet de type de formation, qui est un projet Erasmus+, renforcement de capacité dans le domaine de l'enseignement supérieur, qu'on appelait Mabiova, qui a permis d'installer dans différentes universités marocaines un master dans les agrobiosciences et l'environnement et l'amélioration des plantes. Donc là, vous avez ici les principales universités partenaires de ce projet, donc à savoir de Toulouse qui a coordonné le projet, et Rabat, l'UM6P de Ben Grir, donc l'université de Tanger-Tétouan et l'université de Marrakech. Donc nous avons aussi ici le soutien, nous bénéficions du soutien de l'ENSAT et de AGIREmed pour ça. Donc nous avons ici avec nous Aristide Boucaré qui va se présenter tout à l'heure, qui va donc nous accompagner comme la dernière fois pour le bon déroulement technique de ce webinaire. Donc aujourd'hui, comme je l'ai dit, je vais shifter en anglais pour présenter Tony. I will shift in English, and I wish on behalf of all the organizing committee and also all the attendees here that participate to this webinar, to welcome you, Tony, and to thank you for your involvement and to accept to share with us in these conditions of webinars your work on tomato quality and aroma, and in a context in climate change that you will tell us more to just after. And to introduce more Tony, we call it Tony in our group. It's Antonio Richard Granell. He's a research professor from the National Research Council, CSIC. He's working in the lab that is called IBMCP. So it's an institute of in biologia molecular y cellular de plantas. So excuse my bad Spanish accent. And also, Tony, he's an expert in fruit development and ripening, and he's using for that genetics and genomics and molecular tools. And he's also analyzing natural variability in tomato, and he's developing new tools and collections and varieties by many techniques and biotech techniques to improve fruit quality. He's focused on fruit quality. So the scientific level of Tony he has an H index of 49. He has more than 9,000 citations in more than 150 papers, including Nature papers, Science papers, and so on. And also, Tony is very known in our community as be involved in coordinating European projects. And one of the main of European projects and recent one is one that is dealing with traditional varieties, tomato traditional varieties that we call Traditom, that is a H2020 project. And today, Tony will, he will talk us about fruit quality and the tomato as a model. And just before to give the hand to Tony, just to give some statistics of our webinars. So now we reach more than 1,800 inscriptions, at least to one webinar. So we have 12 webinars that will go until the end of the summer. And we have at least one of these 800 people that have been subscribed to one of this webinar. And what is interesting here that we have young researchers that are mainly following these webinars, that are the master students, maybe from Mabiova and for other master students, and also postdoc and doctorate. And also we have researchers and professors, and also from people from the socio-economic part. And this time, I wish to welcome also people that come from many countries from Africa. We have people from Senegal, people from Burkina Faso, people from Mali that subscribe, and I wish to thank them for their joining. And because Mabiova is, as you can, not Mabiova, AGIREmed association is its goal is to bring people from the Mediterranean and African countries to work together and to join forces to develop agrobioscience and environmental studies. And just to finish and to let more time to Tony to tell us more about his work, just some technical issue here to look. So we choose to switch off the microphone and the video for everybody, just to have a good connection for the webinar. And you can, from now, you can use the button that you have in the down of your window that is called Q&R. It's a French word. It means question-réponse, question and answers. So you can write your questions as soon as you have some question in your mind, and all other attendees can vote on your questions. So the idea here, it's something that this platform bring as facility, is to, by voting on questions, so they would be ranked, and in the end, Professor Smouni Abdelaziz from the University of Rabat will animate this interaction between you and with Antonio Granell by giving you the hand and the microphone to ask your question. And if you don't want to talk, so Abdelaziz will tell the question to Tony. So we encourage you another time, for young people and students, don't hesitate to put your questions, and we'll be glad to give them to Tony and to interact with you. And also, I want to inform you that Tony accepted that this presentation will be recorded, and as soon as it will be available, and after some sanity and polishing, sanity check and polishing, we will make it available for all of you. And also, you can use for all things that do not concern questions, please don't use the Q&R. Use Converser. It's another area when you can ask, maybe if you have problems, you can ask Aristide Boucaré from AGIREmed. You can ask him if you have some problem to listen or to some technical questions, and he can connect with you directly and try to fix it, okay? So please don't put other things in Q&R, just keep them in the Converser part. And in the end, if you want to ask question, you can also use lever la main. You can raise your hand, and so Abdelaziz can see it, and he know that you want to ask question, and also Aristide, he can give you the opportunity to talk to Antonio Granell. So that's it, and another time I welcome you, all of you, and thanks to Tony. And you can now, Tony, share your screen with us and start your presentation. I will just stop mine.
Okay. Let me see if I can do it. Okay, can you hear me then?
Yes, you can put in full screen your presentation. Yeah, thank you.
Okay, thank you, Assis. Thank you, Simo. Thank you, Assis. Thank you, Aristide, and then AGIREmed organizing committee for giving me this opportunity to present the work we are doing in Valencia. As Simo said, it's a small institute located in Spain, by the Mediterranean, and we have like 200 people working there, mostly interested in Arabidopsis and also some crops like tomato and melon. So the title of my talk is Variability in Tomato Health and Flavor Composition and Genetic Improvement. Here you have my email in case you want to interact with me after the meeting, after this webinar, and I will be glad to answer your questions anytime you propose to ask. So my lab is interested in the fruit quality. Fruit quality is a rather loose term, but that depends who you ask, if it's the producer or the consumer or the dealer. It will tell you different things about what he considers a good quality fruit. For us, a good quality, we like to interpret quality traits through the composition of the fruit, because most of the things that we value in fruits have to do with color, have to do with the texture, and have to do with the flavor. And these are traits that are given to the fruit because of the composition, the metabolic composition. In my lab, we use an array of, an array of technologies to address these questions. We use natural variation. We have been able in the last few years to collect and analyze large population of tomato variants or tomato accession, and also wild relatives from the origin of diversification of tomato in South and Central America. We have also constructed our own population recombinant inbred lines on introgression lines that are very good to analyze the genetic basis of traits. And we have developed a number of technologies aimed at identifying the chemical composition of the fruit that, as I said, are the basis for many of the traits that we experience in fruits: flavor, color, and texture. And we have, in this talk, I will mostly center my attention in fruit volatiles, and I will tell you later why. In any case, what we do when we analyze this large population using this technology and also genetic markers, eventually our idea is to identify the genes underlying these traits, these molecules that have to do with fruit quality. We build then maps and try to identify QTLs or regions and markers that are associated with the levels of the different compounds that are associated with fruit quality. The way to do this is to combine the gene mapping with additional technologies like RNA-seq data or gene expression data. Eventually, we confirm the identity of the gene in modifying the level of the specific compound that has to do with fruit quality through reverse genetics. And eventually, sometimes we build models in which we try to model one complex trait like flavor based on the compositions or in the alleles of genes. So this is, in summary, what we do in the lab in relation to fruit quality traits. We mostly work on tomato, but over the years, we have been also working in peaches, oranges, melon, and strawberry. And we have done similar approaches in all these crops. So why to work on fruit quality one, as you were able to listen in last AGIREmed webinar last week by Professor Fossi Bekowi, food production is the most important challenge that breeders have in order to guarantee food security, specifically in the face of a number of challenges like decreasing arable land, increasing population, climate change, increasing pests and diseases. So the breeders, the most important traits for them now are to try to increase and stabilize yield, to increase pest and disease resistance, and increase heat tolerance, for instance, or maximize water use efficiency or nitrogen use efficiency. These are the workhorses of the breeders now. And why then to do something else? Not because apparently this is the most important thing. Well, this is because maybe globally it's important to make sure that we can eat, but for sure in Western country we eat more than enough. So that and obesity, for instance, is one of the most important disorders that many people living in the Western countries experience. And this is just two ways to look at this. It's a map in which the darker the color, the more incidence of obesity. And in all the cases, more than 20% of the population in each of the countries in Europe, and for sure more of that in the different states, United States, are obese. And the reason they are obese is because we don't eat well. We have too much calories, we don't exercise, sedentarism, but mostly not appropriate food intake is the culprit. So there has been a number of campaigns to fight this. And there's a kind of healthy diet that we should follow, in which the basis may be grains. The main focus of productivity, and maybe the talk that you have listened to the last webinar, was how to increase yield. And when you have yield, you always have in mind the main staple food, the rice, the cereals, the grains, highly condensed matter that you then hydrate and eat. But in terms of health, what matters is fruit and vegetables. So, and the reason for that is because fruit and vegetables contain a number of compounds that are necessary for your well-being. And this is not present in rice, it's not present in wheat. So we need these vegetables, otherwise we get a deficiency, like provitamin A deficiency that produces blindness in people who only eat rice or other grains. So, and there has been a number of campaigns over the years, like they eat 5 a day, or go for 2 fruit and 5 vegetables, or just eat more fruits and vegetables. And this campaign basically failed, and it's hard to know why it failed. But one possibility, and this is a general complaint, is that fruits and vegetables, more often than ever before, they don't taste that they used to taste. They don't taste mostly anything. They are tasteless. And this is basic, and tomato has been like a study case for that. The tomatoes the people complain about the loss of flavor of tomatoes. So we thought, well, if one possibility is that people don't eat fruits and vegetables because they don't taste good enough, is we should better look at the basis, the genetic basis or the molecular basis why fruit don't taste good, and try to correct that. So maybe then people will start eating better and get healthier. So, and this takes me to look at what makes fruit taste like what they taste. And of course, different fruits are the compounds that gives fruits the taste, the specific taste is different depending on the fruit, because the fruit don't taste the same. So there are specific compounds that give a specific taste. In the case of some fruits, there is a predominant compound that gives the flavor. And this is the case of pineapple or a strawberry. There are specific compounds, lactones in one case, or furanones in the other, that contribute strongly to the flavor. In the case of tomato, for instance, the situation is not so easy, and there are more than 20 volatile compounds, in addition to 2 sugars and 2 or 3 acids, that gives you the flavor of tomato. So flavor is a combination of sensations that our brain integrate, that come from sensation produced in the tongue basically by sugar and acids, and by the volatiles that are captured in the retronasal part of our head, and then the volatiles interact with receptors, and all this information is integrated in the brain. So there are more than 20 volatile compounds, 2 sugars and 2 or 3 acids that gives you the sensation that you're eating tomato. And the more of the good volatiles you experience, the more you like that tomato. So one important thing is the volatiles in tomato derive from the specialized compounds, but they all derive from primary compounds. So there is the main classes of volatile compounds you find in tomato are derived from branching chain volatiles, amino acids, essential amino acids, or for phenolic volatiles derived for the chemic pathway from phenylalanine and so on, or apocarotenoids that are product of the cleavage of carotenoids, or fatty acid derivatives. These are the main important volatiles in tomato fruit. And it's interesting that, and this is an hypothesis for sure, that these volatiles that the plant uses to attract you to the fruit are all derivatives of essential, for instance, branched amino acid, or important compounds for your health, like carotenoids or other antioxidants. So it looks like, and this is an hypothesis that was raised a while ago by Stephen Gold and Harry Klee, that some of the, in the case of tomato, that the tomato uses volatiles that are derivatives of compounds that are good for your health. And this is a way, maybe a general rule, to reward seed dispersers, because at the end, a fruit is the mechanism that the plant uses to disperse seeds. And so the plant uses these volatiles as a cue for nutrition. So there is an interrelation then between healthy compounds and volatiles that have to do with flavor at that level. This is the list of volatile compounds or compounds in tomato that are indicating that the combination of them tells tomato to your brain. And basically are the 20 volatiles indicated before and the few sugars and acids. How can we learn how the plant builds this bouquet of volatiles that we are attracted to? And is this volatiles change during the evolution of tomato? And then what we did in this case was we use a number of accessions of tomato varieties, in some cases, that represent the domestication and improvement stages. As many of you know, tomato as we know it, this big tomato here, is the product of two steps. One, domestication that took the wild relative of tomato, Solanum pimpinellifolium, into the first domesticated form, Solanum lycopodium theraciforme. This involved mostly an increase in size, and geneticists have been able to map the several genes that are responsible for this increase in size and local number. But later on, it was an additional step, what we call improvement, in which an additional set of genes were responsible for the large increase in size that we find in many tomato varieties. But this has been also this also happened with a reduction in variability. So in pimpinellifolium, there is a large genetic variability that is reduced in the theraciforme and more even reduced in the tomato as we know it. But not only was the change in size that happened during domestication and improvement, there were many more things happening. And this is what I'm gonna show you these slides. By analyzing a large set of tomato varieties, also theraciforme accessions, and a large number of pimpinellifolium accessions, we were able to see how the flavor composition evolved during domestication and further improvement. So when we analyzed the more than 100 volatile compounds in these 600 accessions of tomato, theraciforme, and pimpinellifolium, and we represent the results in a principal component analysis as shown in this slide. All the pimpinellifolium accession map in this region, lower right region of the PCA. The theraciforme map mostly centrally in this graph, in this region, while the large fruited tomato basically spread widely in this graph, but mostly to the left of this. And this I overlay in this type of principal component analysis the loading components. So I overlay now a few, not all of them, of the volatile compounds. And as you can see here, and I coded the volatiles that contribute to flavor in different colors depending on the branch, whether they are fatty acid derivatives and they are in green, if they are apocarotenoids, they are in orange, if they are branched chain amino acid related, they are in blue, and there are phenylpropanol derivatives, they are in purple, and there are other phenylalanine derivatives, they are in red. And as you can see here, the pimpinellifolium, the wild relatives, is relatively, these accessions, pimpinellifolium, accumulate large levels of fatty acid derivatives. When probably man, although there's a recent paper by Caicedo that proposes that maybe the first domestication was not done by humans, but maybe by animals, because it was dated 75,000 years before Christ. That means that before humans inhabit that part of America, in Mesoamerica, or Peru, or North Peru, the Theraciforme accession were relatively rich, are relatively rich in most of the volatiles that contribute to good tomato flavor, including branched chain amino acid, carotenoid derivatives, that with very little of those compounds, you have an impact in flavor. However, in the large fruited variety, these are in the most empty region with no much of the volatiles there. So that means during domestication and further improvement, volatiles have changed. Initially, there was a loss between the pimpinellifolium and theraciforme of most of the hexanal fatty acid derivatives like hexanals and pentanone and hexanals that are characteristic by high levels of those compounds in the pimpinellifolium. Then when we move to the theraciforme, they have huge, very good, high amount of the good volatiles, and most of the good volatiles were lost during during the further improvement. Okay. The good news is that there is still some traditional tomato varieties that are located in that region in this region of the graph. That means that they have more of the good volatiles. So domestication, although initially was size driven, selected by bigger size, also resulted in important changes in the volatile profile and the volatiles that contribute to flavor. Okay. Now, that's what we learned by analyzing this large population of traditional varieties and tomato varieties, and also theraciforme and pimpinellifolium. There has been an evolution during domestication and further improvement. But now we wanted to look what happened in the traditional tomato pool in the frame of this project that Simo was talking about before. Traditon, we analyzed 1,700 traditional varieties from Europe. And we did extensive profiling, metabolic profiling of a subset that we call the core set of varieties that represent most of the genetic diversity in European tomato varieties. And we came up, when we did similar PCA, principal component analysis, based on the volatile composition of the fruits of those several hundred varieties, we end up with this type of graph, in which the varieties spread all over the PCA space. But we can divide this PCA in different sectors, and these varieties here are relatively rich in fatty acid derivative volatiles. So that means that not all the tomato varieties in Europe taste the same, not all the traditional tomatoes taste the same. These ones taste different from that ones here that are rich in branched chain amino acid derivatives, or these ones that are rich in phenylpropanol derived volatiles. And we are starting to do taste panel together with flavor analysis, flavor composition analysis, to see to what extent we are able to distinguish what the machine, the volatile analysis by GCMS machine is able to distinguish. In any case, we have been able to catalog many of the tomato varieties that we find in Europe by assigning whether they have prevalence of compounds, like in the case of Almeria region, there are a number of varieties that are relatively rich in 2-phenylethanol. This compound, although is not a sugar, it gives you the feeling of sweetness. So, and it's a very interesting compound because you can increase the sweetness of a tomato variety by increasing the level of this volatile. This volatile is impacting the retronasal part of your head, of your nose, and you get the feeling of a sweet tomato despite the fact that it has no more sugar. There are other varieties, for instance, in Balearic Island, they have a high level of guayacol, a compound that gives tomato a smoky aroma. In the case of south of Italy, there are a number of traditional tomato varieties that are rich in 6-methyl-5-heptane-2-one, that gives the tomato a fruity flavor. And this is something we learn and something that I think we contribute. But the nice thing about analyzing large collection of varieties that we have resequenced, and this is work we did in collaboration with Samuel Huang and also Harry Klee in Florida, and Danny Samir in Israel. So the nice thing is that if you have this, like in this case, more than 500 varieties resequenced, you have detailed genetic information, and you do extensive volatile analysis, you can combine these two sets and do a genome-wide association study in which you can define what regions of the genome, and here represented in a Manhattan plot, the 12 chromosomes. And whenever you see some dots coming up, this means that this region contains something, a mark, a gene that is associated with high level of this compound. This is MHO, is a apocarotenoid, an important compound that contributes to flavor, and this is geranyl acetone, another volatile that contributes to flavor. And these are the results of 2 experiments conducted either in Florida or in Israel, and analyzed in Florida or in Spain. And as you can see, each of the volatile has a specific pattern with different regions in different chromosomes associated with high level of this compound. We did this for the more than 30 volatile compounds that contribute to flavor, and with also more than 50 other compounds that maybe are regulating the interaction of the fruit with other animals or insects. And we end up with a list of markers or genes that are associated with high level of each of the volatile compounds. What this also could, we could also learn that if we compare Theraciforme here, let's say the closest, the first domesticated form of tomato from Pipinellifolium, to heirloom or traditional, well-appreciated variety, to transitional or modern varieties, and we look at the 3 regions or 3 genes in the one in chromosome 2, in chromosome 3, and chromosome 9, and look at the alleles, we can see here that the recurrent allele, which is the modern one, it has been increasing during domestication and further improvement. And this allele is the one that gives high levels of the volatile. So during domestication and further improvement, the good alleles for high level of MHO have not been lost. Indeed, they have increased during domestication. But this is the exception rather than the rule for the good volatiles and the genes that contribute to high level of the volatiles. Because most of the cases, what happen is that the alleles that are present in the modern or in the transitionals are the alleles that contribute very little. For instance, in this case, RRR or ARRR contribute very little to the level of the compound. So during domestication and further improvement, the good alleles, these ones that give high levels, were lost. Indeed, this is the pinky one here that is present in the heirloom and is lost the further on in the transitional model. While the ones that made it and were selected during within breeding were the ones that give very low level. So, and this is the general rule: the good alleles for good flavor were lost during improvement, because simply breeders have no the tools for selecting these markers, or and they couldn't do GCMS or taste analysis in all the crosses. The rationale we follow for why MHO was not lost while cerealyl acetone was lost during improvement is that both of them are derived by cleavage of isoprenoids or carotenoid precursors or carotenoids that give fruit the colors. For instance, this is prolycopene, this is lycopene, this is zeta-carotene, and this is beta-carotene. So there are specific enzymes that are called carotene cleavage dehydrogenases that cleave these molecules and produces these volatiles. But in the case of MHO, you can produce MHO by cleavage at the position 5'6" or 5' 6' any of these molecules. These ones that are colorless or these ones that are colored, they have color. Our hypothesis that by the farmers or the breeders didn't select for plants that contain high levels of MHO. They select for plants that contain high level of carotenoids because they could follow this trait, high intensity color fruits, and they like that. And by selecting fruits that have high level of the colored carotenoids, lycopene or carotene, they were selecting for high level of the precursor of this apocarotenoid that derive from the cleavage of these compounds. However, geranyl acetone is only produced by cleavage of the non-colored precursors. And because farmers or breeders could not select for fruits that have high level of these compounds because these are colorless, they couldn't follow, then at the end, it was lost by just simple random. This is just an explanation to this specific case. But the idea is that by analyzing variety sets of tomatoes and doing exhaustive metabolic profiling, we are able to identify the regions that are associated and the alleles that are associated with high level of each of the 20+ volatiles that have to do with flavor. And we can now use markers for selecting those lines that contain high level of the volatiles. Another way to study genetically or to introduce perturbation in the volatile network genetically is by making crosses. So in this, and then these crosses can be producing recombinant collection of plants, like in the case of recombinant inbred line. A recombinant inbred line, like in this case we did with moneymaker, Solanum trichopersicum tomato variety moneymaker. We cross it with an accession of Pimpernellifolium that produces a small fruit, very, very fragrant, very with high content of many metabolites. And after crossing, the F1 was cell-fed for several generations using single seed descent to produce 160 recombinant inbred lines. Each line was a stable line after so many cell things that contain a 50-50 combination of the original parents in a chimeric form that is stable. So each line is different from the other. Each line and all of them have 50% proportion, but different regions from each of the two parentals. And these lines are very suited for doing genetic analysis. But at the same time, we realize that each of the lines that we produce a different set of volatiles. So two columns of this is what we call a heat map. And each of the columns is one of the 169 recombinant inbred lines derived from this cross. And each of the rows is one different volatile compound. As you can see, if you look at and compare one column to the next in here, you have almost 160 columns that have different profiles in volatiles. And this is what happened. We generate new variability that is different from the variability we have in the parental lines. These collections that are stable because they have already been homozygous for most of the regions in the genome are very nice to do taste analysis, to do genetic correlation analysis and on other studies. That's what we did. And so when we did this genetic analysis, we identified QTLs, regions that have to do with the accumulation of levels of the specific volatiles. And as you can see here, this is chromosome 1, 2, 3, 4, up and to 12. And then in different, I'm not pretending you can read this, of course, but I have used different colors for each of the volatiles that belong to the same class. So all the pink volatiles, although the pink labels here refer to different volatiles in the family of fatty acid derivatives. The orange have to do with terpenoid, the blue have to do with apocarotenoids. As you can see here in the top of chromosome 1, there is a region that basically has an effect on the levels of accumulation in the fruit of a large number of volatiles. Most of them are fatty acid derivatives, but also members of other families. We now know there is a lipoxygenase C here, for instance, that affect all the fatty acid derivatives, because the lipoxygenase C is a gene at the beginning of a lipoxygenase pathway that eventually produces fatty acid derivative volatiles. But it could also transactivate or transcleave carotenoids. That's why we have also here carotenoids, apocarotenoids that may map to the same region. This is not always the case. There are some chromosomes that are not so rich in regions that affect levels of volatiles, but basically all the chromosomes are involved in one way or another in this population to affect the volatile composition. Okay, so and this information we are using this information in order to change the volatile composition of varieties in collaboration with companies to increase or decrease the level of the good and bad volatiles. Another way to introduce perturbation in volatile composition is by doing introgression line. This is what basically do with retrogressive trait in a varieties by introgression, by crossing and back crossing. But we can also produce populations. Like in this case, we use the same parentals as in the recombinant membrane line, the moneymaker and the pimpinellifolium accession. And here, rather than doing selfing after producing the F1, we did the back cross with the recurrent parent until we produced 16 lines, almost 60 lines, each line representing a small segment of the pimpinellifolium accession in an otherwise moneymaker backgrounds for all the other chromosomes. So here the QTL analysis of what genes are responsible for traits is relatively easy, but because you just have to compare each of the lines with the moneymaker background, because the difference is only in the small fragment that is introgressed. So when we did this with from the line number 1, we were able to confirm that in the top of chromosome 1, there is in pimpinellifolium, there are alleles of these regions that contribute to high level of fatty acid derivatives, but also carotenoid derivatives, apocarotenoid derivatives, and so on. We have been doing extensive work on these genetic mappings of volatile good for flavor. Mathilde Koss also recently published another paper in which she did an integrative meta-analysis using a number of populations, including ours and others, to come up with a similar association, but increasing much more even the number of significant association between sugar acids, amino acid, and flavor-related volatiles, so compounds that contribute to flavor. So when we look at this and see, oh, there are more than 305 loci that affect compounds that are relevant for flavor, this is going to be very difficult to deal with in a breeding program, no? But the good news is that there are more, although that we can target this. There are more than one QTL that affect several volatiles, and by changing the volatile levels of some small number of volatile, we can have an impact on the fruit quality. By comparing all the different approaches people have been using for mapping QTLs, either by GWAS or by biparental or multiparental lines, we end up with this idea that each population gives you information that only in a small number overlap between each other. So the number of specific QTLs in each of the population is much larger than the ones that are shared. When we compare all these with the GWAS results, the Mathilde analysis in metagenomics, or the ones that we did in a collaboration with Haricli, we find that 50% of all these are comprised of the GWAS result using the variability set. So there is plenty of opportunities for breeding, for changing the volatiles. Now I come back to this idea mentioned before, that all the populations bring you opportunities, because, for instance, and because each of them gives you new alleles that provide an extension of the how much you can change the level of the specific volatile. For instance, and I will just highlight the 2-methylbutanol volatile that contributes to good flavor. And in this case, we have in one collection, we have a range of variation that is almost 5,000 times. So we have one variety that has one, and one variety has 5,000 times more than that one. While in other collection that, for instance, this is the collection of heirloom of the traditional varieties, the range of variation is only 60-fold. Because if you want to look for alleles to increase the variability, we should go to this collection of materials here. And this is the case for many others. For instance, in the case of original, the recombinant inbred line is the one that provides the larger range of variation. Okay, so there is plenty of opportunities for alleles that may have an impact on a specific volatiles. But we shouldn't go just for the specific volatile. The good thing is that when we look at the correlation between the volatiles across many populations, we find that they're relatively highly correlated. I showed this here better. For instance, all the phenylpropanoid-derived volatiles are highly correlated. That means if we do crosses to increase one of those, most likely we will get increase in the others. These are, by the way, are the volatiles that at least Western societies don't like in their tomatoes. Eastern society, in the case of Japan and China, they like these compounds in their tomato. So this is also another challenge. You cannot make everybody happy when you want to get the perfect tomato. For instance, the level of apocarotenoid, the carotenoids are also clustered together. That means that if you find a QTL and they increase one of them, they will also change the others. And these are all good volatiles. So if you increase the beta Geranyl or beta-yanon, you will get them together most of the time. So you don't need to increase all of them individually. You can have an impact by cluster, by some of them in the same cluster. Okay, we move now to show you that it is possible not just to have a linked marker that you can use in breeding. You can also go down to the gene in some cases. Here I show you the comparison of QTL analysis done in different populations for a set of volatiles that are called phenylpropanoid-derived volatiles. And in all the cases, in different populations, in the bottom of chromosome 9, we identify QTL for eugenol, guaiacol, methyl salicylate, and ethyl salicylate in different populations. Always the most wild accession produces a decrease in the level of volatiles. This was also identified by the GWAS analysis using the 600 varieties that were resequenced, and this is the chromosome 9 signal that is very important. There was also this, there were papers also by Mathilde and others that showed that in this region of the chromosome 9, there is a QTL for a smoky aroma. In this case, they did, in addition to profiling metabolites, volatile, they also do taste panels. And they used as quantitative trait the response of the taste panel. And they were able to map in this region of chromosome 9, not only the QTL for the phenylpropanoid derivatives compounds, volatiles, but also a smoky pharmaceutical aroma. So most likely the level of these compounds have to do with smoky aroma. And we have been using these markers to do breeding with companies to modify the level of smoky aroma. As I said before, this is a trait that is appreciated in the Eastern countries and not so much liked in Western countries. So when we look at these compounds, the phenylpropanoid derived, the volatiles, like we have here, guaiacol, eugenol, methyl salicylate, and ethyl salicylate, they're all derived from the SCIC pathway. This is a very complex pathway that branches out in different, and these compounds are at the end, almost at the end of each branches. So it could be that the QTL that increases the level of this, that decreases the level of this volatile is a transcription factor that regulates all the steps. This could be one possibility, or could be something that acts up here, but then we should be able to identify all the changes we didn't identify, like in phenylalanine. So, and this is a hypothesis that was raised by Arnaud Bobby and Yuri Tikhonov in Holland, and we started to collaborate with them in this topic. They saw that there was an hydroxyl free group here in all these compounds. And as you know, quite often these groups can be glycosylated. So the hypothesis of the lab was that the smoky, and this was because of the Nobobi and Yuri Tikhonov did extensive, not only volatile analysis, but also non-volatile analysis. When they analyzed the smoky tomato type, they saw that during ripening, the phenylpropanoid emission was constant. There was the smoky release a smoky aroma, smoky volatiles. This was what happened in panel A in this top graph. But when they look at the remaining compounds in the fruit, the ones that were not evolving, there was also these compounds were glycosylated, as this diagram here shows. The compound was glycosylated, so there was some of the phenylpropanoid were emitted by the fruit, but some of the compounds remained glycosylated in the matrix during ripening. But there was release. And the way it was conjugated was with 2 sugars. In the non-smoky tomato type, what they were able to see, looking at the chromatogram, during ripening, the fruit stopped releasing the smoky compounds. But what happened is when they look at the matrix, what was trapped in the fruit was that the compound conjugated to 2 sugars also decreased, but instead it increased another compound for that contained the phenylpropanoid moiety conjugated to an additional sugar or the aglycone. Okay, so the hypothesis is then, and this was what made possible to identify the gene, is that the smoky tomato varieties released the volatiles and conjugated part of it to a glycosylated form. But the non-smoky should have an additional glycosyl transferase, the enzyme that transfers an additional glucose or sugar into the double conjugated form. And this form was not being able to be cleaved by glycosylation when we eat the fruit. Okay, so what Arnaud did was, and Yuri did, was to do RNA-seq data of these fruits and compare to these ones. The hypothesis is the RNA-seq data should contain, in this case, the activation of the expression of a new glycosyl transferase that should be increasing in this non-smoky sample and wouldn't be at all present in the smoky tomato. Also, it should be mapping, and that time the good news was the genome was already mapped, although this region was not so clear, the sequence. They were able to see that in this region of chromosome 9, there was a non-smoky glycosyl transferase. They were also able to see there was, they could be developing a marker that allow us to do breeding with this trait. What we did in our lab, we took the non-smoky glycosyl transferase, produces plant that produced the wild type monomaker, didn't express this gene. When we introduced by transgenesis the the non-smoky glycosyl transferase, there was huge increase, of course, of the transcripts that were not present in moneymaker. And when we look at the volatiles emitted by this fruit compared to the wild-type moneymaker, wild-type moneymaker produces high level of glycol, methyl salicylate, and eugenol, while the plants that contain the new glycosyl transferase didn't evolve any of these compounds. However, when they look at the level of conjugated volatiles, conjugated methyl salicylate, glycol, and eugenol, the plants that the fruits of the plant that contain the new glycosyl transferase have these conjugated forms that were not present in the wild type. So the new glycosyl, non-smoky glycosyl transferase was able to trap the guaiacol, the methyl salicylate, and the methyl and the eugenol in a way that the plant could not cleave and release volatiles. And so the tomato was non-smoky this time, while moneymaker was. So we can target specific genes and get a huge change in the volatile profile. And this is very easy to distinguish. Smoky from non-smoky is very easy. Even almost everybody can distinguish this trait in tomato. Distinguishing all the other flowery, leafy, greeny aspects of the tomato taste requires training. This requires very little training, and it's clear the effect of just one gene in the volatile profile of the fruit. There are other ways than just and in this case, I'll start to play with interaction between healthy compounds and flavor. In this case, what we did here is we took a tomato variety, moneymaker in this case, and as you can see, moneymaker belongs to this class of relatively modern tomato varieties that are relatively pale green when they are green, but they are pale. And the reason for that is they're classified as a uniform ripening. And 90% of the modern varieties, and this was something that we described in the Nature paper in collaboration with Ann Powell, Jim Giovannoni, Alan Bennett, and so in 2012. Most of the, and this was the reason of, part of the reason for the loss of flavor in modern varieties, is that most of them are uniform ripening. This means that they have very amount of chloroplast in the fruit. And chloroplast of the fruit, although in not a big amount, they also contribute to the sugar composition in the fruit, and also to the volatile composition of the fruit, and to the carotenoid composition of the fruit. So it's important to have a large amount of chloroplast in the fruit that eventually will become chromoplast when they are red. So we took this moneymaker, deficient, like many tomato varieties nowadays, in chloroplast in the fruit, and we start to add genes that are able to increase the number of composition of the chloroplast. And we took two of them. One is called GLK2, and the other is called APR2. One that was described by Ann Powell and us in 2012, and the other APR2 was described by the Graham-Seymour group in 2013. And we use that all tomato DNA strategy, all the genes were to tomato, including the promoter, which is an early transcription promoter, and we were able to produce moneymaker derived lines that contain more and more and more of the chloroplast in the green fruit, and eventually more chloroplast in the ripe fruit. And when we look at, for instance, at the soluble solids, the lines that contain GLK and APR2 contain even higher levels of soluble solids. And when we look at the level of carotenoids at the breaker stage of the fruit or at the ripe stage of the fruit, there were large increases in lycopene and beta carotene in single or double combination. And this was good because we increased the healthy composition of the fruit and the flavor. We have more sugars, better taste. We have more carotenoids, antioxidants, or healthy compounds, which is good for food. But we also have an impact in flavor. We have a decrease in fatty acid derivatives and an increase in terpenoids and branched chain amino acids. So, and this was a trade-off, was a consequence of this that we didn't expect. we thought, well, more chloroplast, more of everything in the fruit. No, we get more of some of the components like carotenoids, but we get some of the terpenoids and branched chain amino acids. Some of them are produce intermediates of them, or themselves are produced in the plastic. But interestingly, there was a decrease in the level of fatty acid derivatives, volatiles, which are also contributing to the flavor. So there was a change in the flavor composition that was unexpected. We can also produce changes unexpected changes in the volatile composition by applying new technology like gene editing. In this case, the goal was to target what we call the green flesh gene. Green flesh was identified a while ago by Barry and others in the Gene Giovannoni's group as a small protein involving chlorophyll degradation. As you know, during ripening, the fruit that is in the mature stage is green, becomes red, but during ripening, the chlorophyll is degraded, so that the red comes up very bright during ripening. In this case, these mutants, green flesh mutant, they're a mutant of the type that they describe as stay green mutant. So they don't degrade chlorophyll. The chlorophyll is the molecule that gives the fruit the green shade. Here, these fruits, mutants in this gene, they don't degrade. The gene is now known to encode a magnesium dechelatase that is required for degradation of chlorophyll in all the parts of the plant. So these plants don't produce, they don't yellow the leaves as the leaves get older or get senescent, and the fruits remain green plus red when they ripen. They don't degrade the green chlorophyll during ripening, and the fruits eventually become a brownish color. This more than just an aspect of the fruit that can be distinguished from others, has an added value. Here we target, there are a number of alleles that are present in many traditional tomatoes that are normally called moro, or they are also called dark or fruit, or they have also local denominations, but all of them are loss of function mutations. In this case, we target this third exon for CRISPR, and we produce a series of deletions that basically produce null alleles of this gene. This resulted in fruits that were greener, have more chlorophylls, at breaker +4. At these stages, the level of chlorophyll is very low in the control because chlorophyll is mostly degraded. But in this case, breaker +4 or breakers +8, this is full red already, but the fruits still remain with high level of chlorophyll in the fruit. And this was accompanied with high level of carotenoids, because carotenoids, as you know, get stabilized with chlorophylls and vice versa. So there was higher level of carotenoids in this green flesh mutant that we produced. And we compared the volatile profile. It was the wild type were here, and the different green flesh mutant were in this region, the wild type were here. And then we overload the loadings. We could identify that the green flesh mutants have high level of the apocarotenoids derived from carotenoids, as we expected. And these are very important for flavor. So not surprisingly, these tomatoes that are called, some companies call, and there's a company that call them kumatos, and some of these brownish tomatoes, there are many other companies that also produce those. They are highly appreciated by flavor because these beta-ionon, pseudo-ionon, they act, they are positive, they have a positive impact in flavor, and very little amount of those have a higher impact. Just to finish, I will show you another unexpected result, and this was when you use lines that were enhanced for high level of antioxidant, water-soluble antioxidant. This is work done by Sang and Butelli and Kathy Martin lab. And in this case, they produce tomato lines that overproduce rosé and delilah and accumulate high level of anthocyanin, or MIP12 accumulate high levels of flavonols. I will just show you these two. And as you can see here, when we analyze the level of the volatiles in these mutants that are enhanced for high nutritional healthy composition, what we saw unexpectedly is that they have high levels, much increased levels of beta-ionone and beta-citocitral, two compounds that impact flavor positively. So just to finalize, my take-home message is that during domestication and improvement, there were large changes in flavor composition, not just fruits were selected because of the increase in size. Probably there was also some selection based on composition, based on flavor. We are starting to understand the genetic basis of these changes and how to use this knowledge to improve or change flavor. And also there are many things that affect flavor, including the strategies for enhancing nutritional composition. We have done extensive work also using some of the components of climate change. For instance, if we grow plants in a restricted water, like we expect to have drought associated with climate change, if the water restriction is harsh, then you don't get anything. Normally the plant will shed the flowers, we don't shed fruit. But if you apply limited amount of stress, of water stress, of the salt stress in the roots, then the plant will may limit some of the production, won't be as productive. But the effect on flavor composition is towards in the direction of better flavor. And this is a general practice that is done in many places, where some varieties that not necessarily the best in terms of flavor, if you grow them under limited water condition or under limited stress, not so much as to compromise productivity too much, then you get anyhow an effect on productivity, but you will get an enhanced flavor. And just to finish, this is the people in my lab that works a lot with, in collaboration with me, Diego and Tony, the other PIs and many projects. The work that I have shown here is mostly the work of Jose Luis Rambla, the expert in volatiles, but also Flor did the transcriptional activation for more chloroplast in the fruit. Marta did the engineering for MIP12, and Silvia Giannolio worked in engineering for the green flash mutation. And of course, they have a number of collaborators that over the years have facilitated our work. Gianfranco in Rome, and Giovanni, Gabriela, Andrea Mazzucato, Rafael Fernandez in La Maggiore has been a good friend for many years and a good collaborator. Danny, Harriet, Denise, and Sanwen for the variety set and the resequence variety. Eugenio and Kathy Martin, and on the funding from Tom Gem and Traditor and the partners in that. And by this I will finish. I will happy to take your questions.
So thank you very much, Tony, for this clear and rich presentation that covers many aspects of fruit quality, how we analyze fruit quality, how we improve, going from a classic and traditional method to very, very new methods and up-to-date methods like genome editing, and how it can help to improve, to learn, to understand, and to improve after. So now we have many questions. So it's very nice. So I will now let Abdelaziz Mouni to select one regarding the vote of the public and to start to interact with Tony.
So thank you, Mohamed. Let me first say something. I want to welcome all the participants to this webinar, and thank you for your interest. Mr. Tony Granell, thank you very much for this very interesting and informative presentation. And I want also to say thank you for all what you have done for our students and for what you are still doing for them. Just remember that Tony is one of those who have participated actively to the Mabiova project and to build with us the Mabiova master. So as Simou said, We have many questions. I will ask them according to the rank obtained after voting. So first question from Soufiane Fell, student from the University in Rabat and from Masir institution, who asked why there are modifications and evolution on flavonoids after domestication and improvements.
Well, basically, the level of flavonoids during domestication and improvement has been very little, because the flavonoids in Pimpinellifolium and in Therasiformen and in modern tomato is restricted mostly to the peel and is in the form of naringenyl chalcone that gives the peel the yellowish color. And this is the general trend. Most of the accessions you collect in the wild for Pimpinellifolium will be red fruited with yellow skin. Okay. The only change that is obvious that happened in terms of flavonoids in the fruit that has not been genetically engineered, like the one that I showed by Kathy Martin group, but the natural diversity on the traditional, what we call pink fruit tomatoes that are MIP12 mutants. So plants that do not produce MIP12 transcription factor in the peel, and therefore, or produce very little amounts, and therefore they don't accumulate flavonols in the fruit, in the peel of the fruit. Normally the flesh is almost empty of flavonols, of all the tomatoes, except for the disengineered tomatoes in which we took MIP12 and produced it and regulated, upregulated the gene and the expression of estropropyl. In general, the only effect in flavonols is to almost disappear. If we go, and this is what we call the pink fruit tomato, but by the way, they are highly appreciated in the Eastern countries. They prefer the pink fruit to the red fruit in the Eastern country. And this MIP12 mutation, this work that we did also, someone did, there are almost all the varieties contain the same mutation. They have introgressed in different varieties to produce versions of every sort of variety, but pink, mean no flavonols even in the peel. If the wild relatives of tomato, but very far away from the tomato for domestication, could be Penelli or Chilense, and they accumulate flavonols also in the peel and in response to high UV radiation or a lot of radiation. And this produces accumulation of delphinidine and also anthocyanins. But flavonols as such only in the peel and also only in most of the varieties that are red. Only the pink fruited variety are devoid of that.
Okay, so now we will take a question from the audience, and we have Usama Badat from USA, so we are welcoming him to join us. And I will ask Aristide to give him the opportunity to activate his microphone.
Okay, Usama, you can open your mic. Your mic is already open. You can start to talk.
So much. First of all, I want to thank you for your talk. It's giving answers to probably a lot of my questions in my research right now. I'm doing epigenomic profiling between a wild olive and a cultivated olive from the Turkish Aegean region. And I was hoping for a lot of fatty acid control by the DNA methylation, but I only found out that it's mostly flavor. So terpenoids and linoleic and linoleic acid. I just wanted to ask you a question. Do you have any studies on tomatoes on an epigenomic profiling using DNA methylation? And do you know if there is any control of DNA methylation on the flavors? Because all my pathway, the differential methylated pathways that I found out are linked to flavor instead of concentration of oleic acid, for example, which is the biggest thing in olive oil? Thank you.
Yes, I mean, this is not my field of expertise, but I know the literature a little bit. And what happened, not specifically in the case of flavor. I think what has been reported in tomato is that epigenetics plays an important role in ripening. So there is a DNA methylation of specific residues changing, and this is activating the main drivers of ripening, like green and Nor, and the main mutations and the main genes that regulate ripening. So by regulating by methylation, epigenetic regulation of the main actors in ripening, eventually you will get regulation of the downstream ripening element, like genes involved in flavor, because its flavor is basically built up during ripening, and flavor composition is the same, of course. So, but I'm not aware of specific DNA methylation of a specific transcription of genes involved in volatiles or flavor composition, other than the ones you expect to get when these epigenetic marks are removed from the main regulators of ripening. So everything goes through this main, as far as I know.
Thank you.
Welcome.
Okay. So next question from Q&R section. Question from Professor Zin Labidin Triki from Faculty of Science in Rabat, who asked if flavor traits are monogenic or polygenic.
Well, I think it was clear from my presentation that flavor as such, and we can define flavor in a global sense. I mean, depends how you define flavor. In all the cases, is multigenic. In this pangenomic meta-analysis that was done by Mathilde, the latest one after our publications, and we already reported several hundreds. Here she said that in this unified comparison meta-analysis, more than 300 loci were involved in volatile or compounds related to flavor, in each of them. So, and as I said that also different aspects of flavor may be contributed by different volatiles or with different compounds. So there is like a network. Only we can learn about this with machine learning technologies, not one by one. We cannot decompose the complexity of flavor by analyzing individual volatiles or individual compounds. Indeed, when we analyze fruits that have dramatically different flavor composition, we still see that we don't tell them too much apart. I mean, there's quite an elasticity of a buffering effect in terms for many of the volatiles. So definitely multigenic and very complex.
Okay, thank you. So for the next question, I will join two questions which seem are in the same way. The first one is from Professor Ali Boulerbah from Faculty of Marrakesh, who asked what is the effect of climate and agricultural practices on the flavor composition and quality. And the second one from Aminata Bai, sorry for pronunciation, who asked is that the chemical flavor in tomato depend on the soil?
On the size? I couldn't get the final sentence.
On the soil.
On the soil. Is that the chemical flavor in the tomato depends on the soil?
Yeah, I think well, maybe I don't think there are very detailed analysis on soil composition or I don't know microorganisms living in the soil, or doing people who are very much like the approach of doing not using natural chemical fertilizer, but using natural approaches for cultivation. they claim better flavor, but this has not been rigorously assessed by scientific means. The only thing, as I said before, is that we know that limited stress inflicted into the plants have, when there is, let's say, limited stress that have a small but control effect on productivity, then the most identifiable effect is on good flavor rather than bad flavor. So the sugar composition and some of the volatiles that increase have a positive effect on flavor. So I don't know if the detailed analysis done on changing the effect on the soil, other than this limited stress. I'm not aware of that. Let's say in broad sense.
Okay, thank you. Next question from Julien Pirello from ENSAT INP Toulouse, who asked, do you observe any genetic links between post-harvest conservation traits and volatile compound traits?
Yeah, we have done a lot of this because everybody knows that the post-harvest has a dramatic effect on flavor. And the ways that you normally try to extend the life of the fruit, the shell life of the fruits during post-harvest normally has been inhibiting the levels of ethylene production or slowing the growth of fungi. And this you do either with temperature, basically. In any of the two cases, you have an effect in ripening, or not in general, but in some aspect of ripening. So at the end, what you have is a fruit that doesn't ripen well. And then you have some of the volatiles that do not evolve normally after these shells, after this storage, let's say, perturbations that you inflict in the fruit, and you don't get a fruit that ripens properly. So you get an altered volatile profile and altered flavor. In the case of cold temperatures, that is most of the worst thing you can do with a good tomato, put them in the cold, then the biochemistry more than there are epigenetic changes also occurring in that, but also associated with this lack of coordination in the ripening program that eventually results in a fruit that have a different flavor composition, and most of the good flavor compounds are not present after cold treatment of during post-harvest storage.
Okay, thank you. Next question from Saïd Al-Hasnawi from Faculty of Science, Rabat, who asked if chemical flavor in tomato depend on the biotic and abiotic stresses, and what is the importance of this compound, of this flavor, for the tolerance mechanism through these stresses?
Well, what I know, we know that if you, of course, a plant that has already infected and developing some disorder, they won't develop a good flavor. That's a general rule. But if you introduce disease-resistant genes from wild relatives, because there are so many genes involved in the production of a good flavored tomato, and most of the wild relatives that you use for introducing disease-resistant genes are from varieties that produce fruits that are not even red, but green, and they're not intended for human consumption, you may guess that there are effects on the volatile profiles, and therefore when you introduce disease-resistant genes, you may affect flavor somehow. And we have some indication that this is the case in some cases. Some cases you may not notice the effect, and some others you can detect it by GCMS, and some others you can get effect that you can feel with your senses.
Thank you. So next question from Malika Abu Hussein, who asked about light conditions like intensity, photoperiod, yes, are believed to regulate the synthesis of secondary metabolites.
Yes.
But we know just little information about the effect of light on regulation of pigment and flavor in tomato. She asked, is there any study about that?
Yeah, there are a number of studies but not too much in depth. I mean, and basically, if you consider that, at least in the case of tomatoes cultivated in Spain and many other countries, they are collected when they're at breaker stage. They're put in a lorry, in a truck, completely in the dark, and they ripen along the way. Okay, and eventually, it's not like they are fully ripened on the plant or the truss in sunny southern Spain or southern Italy, southern France. So, and they develop color, deep red color. And of course, you can see differences, and you can fine tune this and find differences, because the more light, the more. But you can get a fully ripe red fruit with no exposure whatsoever to sunny light after being collected and put at harvest in a 12 degrees room without any daylight. So chromoplast after a breaker, its transformation is started, and you can get a nice red fruit. So I don't know much of the detail, not an expert in that, let's say, treating so badly tomatoes, but this is the way we do it. But hardly ever you get a fully ripe fruit from the tree, from the plant, from the truss. And although you can gain some bricks and some amount of carotenoids by prolonging the amount of time the fruit remain in the plant, you can still, it's not so necessarily to get a decent fruit that has been ripened mostly in the dark or very little light.
Okay, so I propose to take 2 more questions, and we will maybe let Tony recover from this meeting. So we have a question from Jaime yes. Provence from UPV. So he's thanking you for this webinar, and he's asking. So Jaime is also a specialist about wild relatives, as you know, and he's asking the following question. He said that the genetic background of modern varieties contains many integrations from wild species. And a recent study from Wageningen shows that around 27.5% of the genome of modern varieties of tomato comes from wild species. Yes. Could this account, at least partially, for the difference in volatile composition of modern varieties and old varieties?
Yes, I think in part I answered this before, but we have results in our lab that points to that direction. I mean, it's something that even without doing an experiment, you can already suspect that this is going to happen. If you have 300 plus regions of the tomato genome that are controlling the level of volatiles, and you introduce 20% of the genome of wild relatives into the tomato, and you cannot pretend not to change the status quo of the volatiles in the traditional tomato that are devoid of those. So for sure, yes, in a more or less extent. Then the thing is that normally breeders have, of course, the first thing is to get a plant that can resist the disease. And this is the main important thing. You cannot have flavor with no fruit, no plant. So this is something we need to do. And the problem with these broad crosses is that even if you screen many plants, it's very difficult to get introgression that are smaller than a certain amount, specifically in some regions when long crosses, you produce long crosses that have very low recombination. At the end, you carry over this 20+% when you have several disease-resistant genes introgressed. And of course, you expect to have an effect, and we know that we have effects. Whether these effects can be important for making the tomato not palatable or not good to eat, for sure not, because otherwise they won't sell it, but whether they could affect negatively the good flavor that people claim all tomatoes may have, yes, for sure.
So Aziz, I propose that you take a last question from the Q&A.
last question. We have one from Mohammed Nahiri from Tangier, who asked if strigolactones derived from carotenoids play a role in the flavor.
Oh, no idea. I mean, the people who does this strigolactone normally works in roots and very specific models. I'm pretty sure it's going to be very difficult to detect the strigolactones in the fruit with the current methods, not in a high throughput way. So I'm not aware of anybody working at that level. I'm sorry, can't help. There are plenty of carotenoids in the cow, in the fruit. So I don't know how, my guess that there is the strigolactone will be minor contribution, but I cannot say.
Okay.
Maybe one last question, which can be interesting for consumption of tomato. Mr. Ali Boulerbah, who asked, what is the effect of cooking on the flavor composition?
Oh, no, cooking is something that changes dramatically the compounds. I mean, because there are many reactions that occur between amino acids and sugars. It is like adding the spices, of course. Then it's a new world. This is for food technologists. What I know is that we hardly get more than 100 compounds when we do with the new machines, GCM machines. I remember reporting with they use both fresh tomato and cooked tomatoes to see how many compounds we can get, and there were more than 500. So maybe this explains that we get more compounds that normally adducts or products of the reactions at this high temperature between the compound that you will get in a fresh tissue. After cooking, you get lots of new compounds by reaction. But in this term, probably one order of magnitude, maybe more, or who knows. I mean, the report claimed 500. We, with the new machine, we hardly get to know 100. So this is good explain. But I'm pretty sure cooking, or depending on what type of cooking you do, especially with oil, that will increase the temperature a lot. You will get very much a dramatic change in composition.
So
you I have
no more cooking tomato.
No, different. It's a different thing.
salad.
Because according to were introduced for cooking, but I mean, cooking raw tomatoes were not the first thing the humans really thought about.
Thank you.
So I have two last questions,
Yes.
very quick ones. One that is related to what we just said, and the last one will make a link with the next webinar. Okay. So the first one, I know Mathilde Kos is with us, attending the meeting, and she repeats this in many places. And do you recommend or not, or you do not recommend to put tomato in cold in the freezer? Okay,
This is
to conserve them. Yes.
I mean, if you cannot eat them before it gets rotten, better to maybe it is tasteless.
Yes.
Because if you're going to use it for cooking, maybe it's not that important. For salads in which you want to appreciate the good flavor, better keep it out of the fridge.
Yes. So there is an impact of the cold for conservation on the flavor. Okay. So the last question is. What do you, we had some question about the effect of soil on aroma.
Yes.
So what is your opinion on the effect of the microbiota of the soil on the aroma?
I'm pretty sure that there will be lots of interaction and probably people, but unfortunately, I guess that 90% of the production of tomato is done in almost sterile conditions, in soils that contain very little microbiota, is sterile coconut fiber, all that kind of substrate that are very much different from the ones in which you use manure or green manure. The population of microbiota in those cases are huge. What is the effect? I'm not aware of anybody doing serious work about this, because probably it's very difficult to do, because how you control the microbiota? I mean, this is like say, okay, we do traditional cultivation using manure, and we use chemical fertilizer in almost sterile soil. My first, I would say that it's better to have this in manure, maybe, but this is normally associated with people who grow tomato seasonally, like with traditional tomato, different varieties. And of course, this is very difficult. We haven't done the experiment. If somebody has done the experiment, maybe they can contribute, but we haven't done the experiment in parallel, using the same varieties. Normally, the varieties they say they have been developed for optimal growth in greenhouses under very controlled conditions, using chemical fertilization and recovery of the irrigation waters, and it's different thing. And so I don't know of a rigorous study trying to compare these two things.
So this makes the link with the next presentation by Professor Mohamed Hijri, that was leading a lab in Canada, and now he's a professor in the UMCC University. So he's a specialist of microbiome and microbiota, okay, on plant, and he have a very nice record of papers, and he have a medium of one Nature, one Cell per year on this subject, and so he's very specialized on this. And he's actually giving a webinar in another part on this, and he's also involved with us, and he will give the next webinar, and he will talk more about the effect of microbiota on plant. So I encourage you to, all of you that can attend, so it will be also a rich discussion, and on behalf of all the management committee of AGIREmed and all the participants today, we thank you another time, Tony Granell, and we are very happy to have you. And also now the students of Mabiova can see you.
Yes,
they are following your courses on the digital platform on Moodle, and now they can see you.
Thank you.
And interact with you, and thank you again, and we continue to work together and in the frame of this network. Just to finish, I don't know if you know it already. I think that European Commission is liking your work very much. So we just received an email that saying that they like your work.
Okay.
You will see what I mean with this.
Let's see.
and congratulations for Harnstorm, Tony.
All right, thank you all.
Okay.
Thank you, Tony. Thank you very much.
Keep safe.
Yeah, with this good, with this good news, and maybe there's good waves with the AGIREmed.
Yes.
And so let me, as I said, on behalf of all people here, to thank you, and let's continue with the next webinars to interact and to exchange knowledge between us. Thank you very much.
Thank you. Bye.
Bye-bye.
Thank you. Bye.
Bye.
Thank you, Tony.